Welcome to Roadmap to Residency, Evidence-Based Applications. So this is a very short video about antigen processing and recognition. We have talked about MHC, compatibility complex molecules in the previous videos. So in this video, we will just be targeting the antigen processing and how antigen is recognized. So T cells, so there are two main types of T cells. They are the CD8 plus and CD4 plus T cells, which are CD8 positive and CD4 positive, obviously. So the cytotoxic CD8 positive T cells kill the intracellular organism, that is mostly viruses and some bacteria. Its T cell receptor binds to the MHC1 on all nucleated cells, and once activated, it will release the cytotoxic granzymes, granulolysins, and perforins, which causes apoptosis of the target or infected cell. The CD4 plus T cells have many types. The TH0 or TH0 is an upstream CD4 plus T cell subtype, and this can differentiate it into, most importantly, TH1 and TH2. The TH1 primarily activates the macrophages. The TS2 primarily stimulates the B cells, enabling antibody production. Now, when a cell loaded with antigen or on MHC1 or 2 comes into contact with a CD8 plus T cell, also called as cytotoxic T cell, or CD4 T cell, which is also called as helper T cell, respectively, the primary signal is merely the binding of MHC to both TCR and CD molecule. In other words, the MHC1 binds to the TCR positive CD8 cells, and MHC2 binds to the TCR positive CD4 cells. So a secondary signal then ensues, that is the CD80 or CD86 on the nucleated cell or antigen presenting cell binds to the CD828 on the naive T cell. And then the T cell is now activated. In other words, the second step is the CD8 or CD86 binds to the CD828. And this second step is the same for both CD4 positive and CD8 positive T cells. And then the activation of T cell that has happened here will result in CD8 positive cytotoxic cells directly kill the nucleated cells that have antigen loaded on MHC1. And CD4 positive T cells are able to exert a variety of effects depending on the differentiation. So we have taught that there are multiple of CD4 positive type of T cells. So the activation of the CD4 positive T cell usually occurs after this differentiation. So the TH0 can become TH1, 2, and 17 and regulatory cells. The TH1 cells stimulate the macrophages, which kill phagocytosed pathogens if necessary, and organize into granulomas. The TH2 cells stimulate the B cells to become plasma cells, which make antibodies that kill extracellular pathogens. And the TH17 cells stimulate the neutrophilic production and recruitment. And then the T regulatory cell prevents autoimmunity. You can see that they are a suppressive type of T cell. So here we can see that the pathogen or extracellular antigen is phagocytosed by an antigen presenting cell and placed into a vesicle. The ingested pathogens are digested by lysosomes to extract their antigens. And then the antigens bind with MHC proteins, right, that enter the vesicle. And then the MHC proteins now carrying antigens are released from the vesicle and travel to the outer surface of the cell membrane. And then the dendritic cell is now presenting antigens, which will activate the T cells that bind with the MHC proteins. So antigen acquired from inside the dendritic cells, antigen acquired from outside the dendritic cells here. And then there is the antigen presentation. So the antigen processing for MHC1. So the partly folded MHC class 1 alpha chains bind to the calnexin until the beta-2 microglobulin binds. Okay, we have seen that MHC1 have the substrate protein beta-2 microglobulin, remember? Now the MHC class 1 complex is released from calnexin and binds a complex of chaperone proteins and binds to a TAP via tapacin, okay? TAP via tapacin. And then the cytosolic proteins are degraded to peptide fragments by the proteasome and a large multicatalytic protease. And then the TAP delivers a peptide that binds to the MHC class 1 molecule and completes its folding. Now, this fully funded MHC class 1 molecule is released from the TAP complex and then it is exported. Extracellular proteins are processed in endosomes by hydrolytic enzymes for MHC2. So the antigen is taken up into intracellular vesicles. In early endosomes of neutral pH, endosomal proteases are inactive. So the acidification of vesicles activates the proteases to degrade the antigen into peptide fragments. And the vesicles containing peptides fuse with vesicles containing MHC class II molecules. Talking about the T cell and B cell activation. So the antigen presenting cells ingest and process the antigen and then migrate into the draining lymph node. Here you can see the antigen presenting cells now have ingested the antigen. Now then there is the T cell activation, single one. T cell activation, the exogenous antigen is presented to MHC2 molecule, here, MHC2 molecule, and recognized by the T cell receptor on the TH cells, okay? And then there is the proliferation. And survival. That is the co-stimulatory signal via interaction of B7 molecule. Here, this B7 molecule on dendritic cell and CD28 on naive T cell. Then the activated T cell axons come into action. Talking about the B cell activation and class switching. So the TH cell activation is as above. After that, the B cell receptor mediated endocytosis takes place. Okay, the B cell receptor mediated endocytosis takes place. And then the exogenous antigen is presented on MHC2 and recognized by TCR on TH cell. Then the CD40 receptor here, the CD40 receptor binds with CD40 ligand on T-helper cell. So this is the T-helper cell and this is the B cell. Now the CD40 ligand of the T-helper cell binds with the CD40 of the B cells. And then the TH cells secrete the cytokines, okay, here, cytokines that determine the immunoglobulin class switching of B cells. And then the B cells are activated and produce IgM and they undergo class switching and affinity maturation. So the binding of antibodies to antigens inactivates antigens by neutralizing, that is blocks the viral binding sites or coats the bacteria, agglutination of microbes, precipitation of dissolved antigens, or activation of complement system. Now, these three can lead to phagocytosis, and the activation of complement system can lead to cell lysis. So the role of an antigen-presenting cell is the phagocytosis of enemy cell, that is antigen, fusion of lysosome and phagosome. Enzymes start to degrade enemy cell broken into small fragments, and the fragments of antigen presented on APC surface. The leftover fragments are released by exocytosis. So that's the process here. So MHC2 binds to the T cell receptor and CD4 expressed only an antigen presenting cell, have two proteins alpha and beta, and they must merge with acidified lysosome. Thank you for watching. Keep studying.